has a full complement, a full genome, and it starts dividing. So you have a fertilized egg. It's one cell with a full genome, 46 chromosomes. And it divides into 2 cells with 46 chromosomes each. Divides into 4 cells with 46 chromosomes each. And it keeps dividing and dividing and dividing until you get a little thing that looks like a raspberry. It's a ball of cells. And every cell is identical. They all have 46 chromosomes. How does that little ball turn into fetus Judy? It's got to turn into different types of cells. So all those identical cells of the blastula have to differentiate. Some cells have to turn off the genes to be blood cells or skin cells and turn on the genes to be nervous system cells. Other cells have to turn off the nervous system cells and the blood cells and turn on the skin cell genes. See what I mean? So when that little ball of cells starts turning into a fetus, it has to turn into different kinds of cells. So some cells have to turn into nerve cells, some cells have to turn into skin cells, some cells have to turn into blood cells, muscle cells, whatever. Each type of cell has to differentiate. And how do cells differentiate? That's a big question for scientists. So operon theory was one of the early models that helped us begin to perhaps understand what's happening. A cell can turn on expression of a certain gene, and it can turn off expression of a certain gene. Okay? That's what we think. What makes this cell become a skin cell and that cell become a blood cell? This one turned on genes and turned off other genes. This one turned on other genes and turned off those other genes, right? So, operon theory is an early model from bacteria. We can understand regulation of gene expression to a certain extent, and maybe that helps us understand how cellular differentiation happens. But operon theory, we're going to do it really simplified. First of all, there are 2 general types of enzymes. Now, actually, there are many, many types of enzymes, but we're just going to generalize. Some are called constitutive. They're major constituents of the cell. They're essential to survival. Therefore, they must always be available. It's programmed into the genome at what rate they'll be made, and there's no way to change that. Other type of enzymes we're going to call non-constitutive or regulated. These enzymes are subject to regulation of gene expression. what do you want to remember for your test? Constitutive enzymes cannot be regulated. It's already programmed. They're going to be available. They must be there. What if I had a mutation in a gene for a constitutive enzyme? Probably going to die. If I don't get the essential enzyme I need, probably going to die. That could be a bad one. Non-constitutive enzymes are subject to regulation. How do you regulate manufacture of an enzyme? You regulate the gene. If a gene is expressed, that means the gene is turned on. The gene is opened, the mRNA can be made, and it can be translated into a protein. If it's not expressed, the gene stays closed, mRNA cannot be made, no proteins get made from that gene.
Is that like it's either off or it's on, like a light switch, or is it
No,
like
two different mechanisms. Two totally different mechanisms. So that's why I put it this way. Some genes can be some genes can be repressed, and some genes can be induced.
but there's no like halfway on, like so many enzymes can be made, but not too many?
Well, not in these particular cases. What we'll talk about that. What regulates how much is made is the presence of the substrate, which indicates need. So, remember back in chapter 5, the metabolism PowerPoints? They talked about a feedback inhibition as a way to regulate enzymes. This is actually what they're talking about. I don't like the placement of it in this textbook, but whatever. All right, so constitutive enzymes cannot be regulated. Non-constitutive enzymes or regulated enzymes can be regulated. Or you might think of the gene as being regulated. So what are we regulating? Manufacture of an enzyme. Manufacture of an enzyme is gene expression. If the gene is expressed, the enzyme that it codes for is made. If the gene is not expressed, the enzyme that it codes for is not made. So expression means the gene is open, mRNA is being made and translated into a protein or an enzyme. Okay, let's talk about repression and induction. I don't know what I want to erase here, but okay. Actually, kind of simple. I like to start with repression. In repression, you're going to shut off expression of a gene. Let's do it this way. Shut off a gene that normally is expressed. How are you going to do that? You're going to do it by feedback inhibition. An accumulation of end product is going to pile up. And when it does, it's going to signal the promoter and tell the promoter to shut off the gene, because I don't need the product of that enzyme anymore. So in your textbook, they talk about the lac operon. We're going to do it simplified. Okay, let's say there's an enzyme right here. This codes for lactase. Lactose is milk sugar. If milk sugar is present in my environment, I'm going to make a lactase enzyme, and I'm going to digest it into glucose and galactose. And that's what I'm going to use as my food source. That's going to go to my cells and be broken down and make ATP and blah, blah, blah. Okay, let's say I'm a little bacterial cell and I live on a dairy farm. So on a dairy farm, I very frequently eat lactose. So I usually make lactase. That gene is always expressed for the most part because I'm always making lactase because I'm always eating lactose. I break it down, I get my glucose, lactose, I get my fill, I'm good. One day, Sloppy Farmer kicks me out of the cow patty into an entire bucket of milk. And in that bucket of milk, I'm cranking out the lactase and I'm digesting lactose and I'm so full of glucose and galactose, I got it coming out my ears and mouth. It's spilling out oatmeal. Stop, somebody stop me please. Do you ever feel like that at Thanksgiving? You're like, somebody just push my chair away from the table because if I eat any more, I'm gonna spill. Stop the insanity. That's what repression is. I have so much of this that I keep making the enzyme and breaking it down to this. Now I have so much of this, I can't stand it anymore. So I have to stop making this until I use this up. Once I use this up, how long does it take you to get over Thanksgiving dinner? Well, maybe a day. And the next day you're ready to go back for the leftovers, right? Mm-hmm. So you pause, you stuff yourself, finally decide to stop the insanity, stop digesting it, stop eating, use up what's already in your full belly, then after a day or so you've used that up, now you can go back to digesting more. That's repression. Repression is shutting off a gene that normally is expressed. And it's a feedback inhibition. It's an accumulation of end product. The accumulated end product actually has an active site